Okay, so my students are doing the lac operon, and that is, they're going through the process of doing gene regulation, whether gene is turned on or whether gene is turned off. So they're using gene regulation for prokaryotes, and a real life simulation is the lac operon, because everybody has E. coli in their intestines, and the job of E. coli is to, if you drink a dairy product, or if you have a dairy product, that E. coli is responsible for being utilized in breaking down lactose. So their job is to go through the process to see if lactose is present, what do they need to do, and if lactose is absent, what needs to be done. So it's taking us back from what we already studied from protein synthesis, what they already know about genetics, and they have to be able to connect all the information together in order to understand this. And then after this, we're going to go into biotechnology. And my students are working in groups of 2. I'm just here as a facilitator, and that is pretty much it. Okay, so they have their student guide, and then they have their lac operon, which has it already labeled regulator, promoter, operator, and the different genes. And then they have their RNA, this simulates RNA polymerase, and this simulates, I believe, going to be like their operator. And then they have their enhancer, which is going to be our inducer, whether it's going to be turned on or off. So they have to go through the process and do both. For the lac operon, they have to do it whether lactose is present or if lactose is absent. They have to go through both models, and then as a group they have to answer questions that go along with the student activity, which, you know, it could be represented as a lab, but I consider it just to be a student activity. And then they have to answer some College Board questions for AP Biology that goes along with it so that they're prepared for it on the AP exam.
What are those?
These represent genes. These represent like lac. This is considered to be lac Z. This is considered to be lac Y, and this is considered to be lac A. Whenever you combine them together, they're responsible for going through and making messenger RNA, which is going to be responsible for coding for a particular protein that E. coli needs to break down lactose in your digestive tract. If you got this white square and you got your yellow square and your triangle, you know what it is, right? So, all right, what you need to do.
Blue and
Can you use a pen?
Which one? Oh, okay. Since RNA polymerase is not, it cannot transfer glycogen back to the genes. The operand is now turned on lactose is back. What happens when lactose is once again present and the cell needs to utilize it for energy? How is the repressor when lactose is again present and place the colored pieces of paper back under the operand. Is that this? Wait. Oh, okay. Push the lactose paper clip through the slit and the holes in the repressor and put the repressor off the operand. What? Right here, step 8. Doesn't it already have it? You grab one without that? There was supposed to be a hole.
Make sure you write your name on the back of the yellow, orange, white square. On the back, write your name on the back.
Okay.
gotta draw one that's going with, okay.
Boom, turned on.
On and off.
Do it matter which side? Like, is it inducible?
You repeat my question? Oh, I asked them how do we show it? How do we show the light operon being turned on?
So how do you figure?
Huh?
Okay.
Because that's what it's asking us to do. So we have to draw the RNA polymerase on the light operon.
Which is code for on the lab glycan allows lactose to be transported into the cytoplasm. Is that it?
Or we can just put it, go here, you see, it says RNA ribosomes.
Yeah.
You can do both.